The hyperphosphorylation of the RB protein is often used as a molecular correlate of cell cycle commitment and distinguishes G0 from G1 cells. We see a very clear delineation along this central arm between cells with hyperphosphorylated RB and cells with hypo or unphosphorylated RB. Now, this dividing line provides a very clear visualization of what is referred to as the cell cycle restriction point on this landscape. It allows us to separate G0 from G1 cells on our structure. So we also used a random forest model to predict cell cycle age using the tracked and annotated data. And we very clearly see an increase in age, both as cells progress through G1, S, and G2, but also along this arm of G0 cells. So altogether, we have a pretty clear idea of how to interpret the cell cycle landscape, which seems to be made of two main trajectories. We have a cyclical proliferative trajectory starting along the central arm and progressing along the right arm through G1, S, and G2 before looping back to the central arm during mitosis. And along this leftmost arm, we have an arrest trajectory with cells spending a longer and longer amount of time in this arrest state as they progress up this arm. I'd like to also point out that we obtain the same general structure over a very wide range of hyperparameters in fate, which is really nice to see. And we also find a remarkably similar topology when we perform the same experiment in a completely different cell line, these HPNE human pancreatic epithelial cells. So these are all very promising signs. So we next asked if we could capture known cell cycle biology on this landscape. And so we took a look at whether we could visualize the molecular mechanisms that control the transition from G1 into the S phase. Now the core unit regulating this transition is the inhibition of E2F transcription factors by the RB protein. Relief of this inhibition and commitment to DNA replication is driven by the phosphorylation and inhibition of RB by cyclin CDK complexes. Now, in early G1, RB phosphorylation is performed by cyclin D CDK4/6 complexes, and we see that most cells begin their cell cycle with high cyclin D. Now, this cyclin D-driven inhibition of RB in early G1 initiates E2F-mediated transcription, which we can also detect since E2F stimulates its own production. Another important E2F gene is cyclin E, which begins to rise in late G1 as E2F-mediated transcription ramps up. And by forming a complex with CDK2, cyclin E helps compensate and maintain RB phosphorylation as cyclin D levels begin to drop. Another important event in the G1S transition is the inactivation of APC complexes, which act to degrade and prevent the accumulation of S-phase proteins. So this increase in cyclin E CDK2 activity in late G1 also stimulates the destruction of the APC subunit CDH1, thus allowing S-phase initiation. The inactivation of APC also allows another important S-phase gene, cyclin A, to accumulate as S-phase begins. Now, cyclin A CDK2 complexes then take over the RB phosphorylation duties as cyclin E levels begin to drop during S-phase. And of course, we see the most important event of S-phase, DNA replication, with DNA content doubling over the course of S-phase. So altogether, this demonstrates that the topology captured in our cell cycle landscape not only incorporates, but is actually shaped by the molecular mechanisms that govern progression through the cell cycle. Now, as I mentioned earlier, much of what we know about alternative cell cycle trajectories comes from work studying the fate of daughter cells following mitosis, with some cells immediately re-entering the cell cycle and others spending a brief stint in G0. So if we highlight the very youngest of our cells, we see a very clear bifurcation of cells following mitosis into either G1 with high RB phosphorylation or into G0 with low RB phosphorylation. The cells that head down this arrest trajectory also show an increase in the expression of P21, which induces RB dephosphorylation through the inhibition of CDKs. If we plot out RB phosphorylation by cell cycle age, we also see that all the cells seem to finish mitosis and begin their cell cycles with high phospho-RB, and then diverge into two populations. One that maintains high RB phosphorylation and enters G1, and another where RB phosphorylation dramatically drops and cells enter a state of arrest. Now, these cells are also colored by P21 expression, and we see that this drop in RB phosphorylation and entry into G0 coincides with an increase in P21 in these cells. Now, while this seems to be one way that cells can exit the proliferative cell cycle into a state of arrest, we also see some indication that cells expressing high P21 in G2 might exit the cell cycle prior to mitosis and enter a state of arrest further down their arrest trajectory. Now, unfortunately, since this experiment was performed on cells growing under ideal cell culture conditions, it wasn't necessarily designed to get good coverage of these arrest states. So it's hard to make any real solid conclusions about these arrest trajectories from this data alone. So to better explore the topology of cell cycle arrest, we performed another 4-eye experiment after subjecting cells to a variety of stresses known to induce cell cycle arrest. So first we subjected cells to hypomitogenic stress. In other words, we grew them in serum-free media devoid of mitogens for 1 or 7 days. I would like to point out that once again, we obtained a very similar trident-like structure in this experiment in control cells, consisting of a cyclical proliferative trajectory and an arrest trajectory extending from this middle arm. We see that the majority of star cells reside in this arrest state with low phospho-RB and with 2C DNA content, indicating that these cells exit the cell cycle prior to DNA replication. Now, this exit from the cell cycle seems to be primarily driven by a decrease in cyclin D expression relative to control cells, which is consistent with the fact that mitogenic signaling induces cyclin D expression, normally induces cyclin D expression to promote CDK activity and RB phosphorylation during G1. Now, the majority of these starved cells diverge from the normal cell cycle in G2 and undergo mitosis directly into this starved state, into this arrest state. We also see a proportion of cells that seem to undergo normal mitosis and then exit the cell cycle from the subsequent G1. In fact, this seems to be the trajectory that the control cells take en route to this arrest state. However, as you can see, we observe very few control cells that end up in this terminal mitogenic state, which is a hypomitogenic state, which is consistent with the fact that these control cells here were grown in the presence of serum. So that should maintain sufficient mitogenic signaling and cyclin D expression in G1. So we don't actually expect, and of course we didn't see this before in the previous experiment, too many control cells to end up in this hypomitogenic arrest state. In fact, as we saw previously, this arrest state in most control cells seems to be instead be driven primarily by an increase in P21 and not a decrease in cyclin D. And so progression further into this hypomitogenic state is actually driven instead by an increase in a related CDK inhibitor, P27. So in summary, hypomitogenic arrest is a single arrest state and is driven, it seems to be accessible from 2 distinct trajectories of cell cycle exit. And is driven into this arrest state by an increase in cyclin D and is stabilized by an increase in P27. Sorry, is a decrease in cyclin D and stabilized by an increase in P27. So next we assess the effect of replicative stress on cell cycle progression. And here we treated cells for up to 4 days with etoposide, which is a chemical that prevents the religation of DNA during replication to induce DNA damage, specifically in S phase, which basically mimics the effect of natural replication stress. We observed that etoposide induces 2 categorically distinct states of arrest with either 2C or 4C DNA content, indicating that in response to replicative stress, cells can exit the cell cycle either from G2 after DNA is finished, but before mitosis, or from G1 after cells have completed mitosis. So consistent with this mechanism of action, we also observed that both of these states of arrest are driven by DNA damage signaling. First, we see that some cells can divert from this proliferative cell cycle here in G2, driven by an increase in DNA damage, measured here by gamma H2AX. Also, an increase in the activation of CHEK1, which is an important component of the DNA damage response machinery, and an increase in the expression of P53 and P21, which is a downstream transcriptional target of P53. We hypothesized that cells that don't accumulate that much DNA damage in S phase after etoposide treatment, maybe because they were in the late stage of S at the time that we hit them with etoposide, do not mount a sufficient DNA damage response in G2 and actually continue through to mitosis. However, as has been shown previously, daughter cells can retain the memory of that DNA damage from the previous cell cycle and increase P21 expression in the subsequent G1, causing cell cycle exit into this post-mitotic arrest state with 2C DNA content. Now, if you remember from the first part of my talk, this seems like the same trajectory that we propose for control cells as they exited the cell cycle after mitosis. And we also see that the majority of arrested control cells in this experiment reside in this arrest trajectory, consistent with these cells incurring a mild natural replication stress during their previous S phase, and that this is why they're exiting the cell cycle. Now, if we take a closer look at the arrest trajectory coming out of G2, we notice that it's actually a bit more complex than I've described so far. So what's going on here? If we look at the expression of cyclins that are normally expressed during S and G2, we see that cells exit the cell cycle with high levels of both cyclin A and cyclin B, and then abruptly shut them down as they progress further along this trajectory. Now, normally, the degradation of these cyclins is driven by mechanisms that occur during mitosis. However, we observe no signs that cells are actually dividing along this trajectory. And most clear is the fact that they retain 4 copies of their chromosomes instead of dividing them into 2 through mitosis. This is actually an example of a cell cycle phenomenon known as mitotic skipping, where cells don't even make an attempt at mitosis and instead transition into a molecular state that's more akin to a G1-like molecular state. In fact, we see that after the S and G2 cyclins are degraded, we see an increase in the expression of G1 cyclins, cyclin D, and cyclin E. And it's been reported that cells can, in fact, re-enter the cell cycle from this arrest state. And if you look along this trajectory here, we see that cyclin D first increases, followed by a later increase in cyclin E, consistent with the normal timing of cyclin expression as they progress through G1 towards S phase. In fact, we also see along this trajectory a progressive increase in CDK4 and in the DNA licensing factor CDT1, all events that should promote entry into S phase. So let's take a closer look at this region. We see that cells progressively populate this putative trajectory of cell cycle reentry over time following etoposide treatment. And another piece of evidence that the cells are re-entering the cell cycle from this state is that we see polyploid cells with greater than 4C DNA content start to appear after 3 or 4 days of etoposide treatment, consistent with cells in this 4C arrested state re-entering S phase, re-replicating their DNA before skipping mitosis and increasing their ploidy once again. And finally, we have oxidative stress. Now, the dominant cell cycle effect we observe in response to oxidative stress seems to be primarily related to its known ability to induce DNA damage. Similar to etoposide treatment, we observe cells exiting G2 with high levels of DNA damage and entering a 4C state of arrest. We also observe cells entering the 2C arrest state directly from G1. However, one major difference between oxidative and replicative stress is that we don't observe any polyploid cells here in response to oxidative stress. Now, this is consistent with hydrogen peroxide actually having a very short half-life when added to cells. So unlike etoposide, which should continue to induce DNA damage even after several days if cells were able to find their way back into another S phase, causing perpetual DNA damage, mitotic skipping, and polyploidization. Presumably, cells that re-enter the cell cycle after the peroxide is metabolized are able to resume normal mitotic division and maintain ploidy from that point on. So if we put all of this together into a single landscape, it is clear that cell cycle arrest is actually a complex architecture of molecular trajectories. We find that different cellular stresses can induce different arrest states, but we also find that single stresses can induce multiple arrest states within the same population. We also outlined this mechanistic trajectory through which sustained DNA damage can induce polyploidy through mitotic skipping. Altogether, through a completely data-driven approach, we're able to generate this structural representation of the cell cycle that both captures known biology, but also reveals a more complete description of cell cycle topology, complete with both proliferative and arrest trajectories. So with that, I'd like to thank Jeremy and the rest of the Purvis Lab. In particular, I'd like to thank Kasha, Tarek, and Colin, who assisted on this project very much with the computational aspects of it. I'd also like to thank my army of undergraduate experimentalists, Holly, Sophie, and Catherine, who helped produce a lot of the data that you saw today. I'd like to thank our collaborators in Gene Cook's lab here at UNC. I'd like to thank you for your attention, and I'm happy to answer any questions.
Thank you, Wayne. And that was wonderful. I very much enjoyed your presentation and have a number of questions. But let me first start with a question that Gabe Boyle asked. Is there a reason why you chose RPE cells versus other non-cancer cell lines?
Yeah, I mean, it's a little bit of an arbitrary choice. We use RPE cells a lot in the lab to study the cell cycle. RPE cells in general are used a lot to study the cell cycle in other labs. and we know a lot about the cell cycle of RPE cells, so it was easy. We figured it would be easier to make sense of the data that we would get following the 4i.
Mm-hmm. And a question that I have is about your selection criteria for the set of proteins that you imaged. How did you choose them? And the related question to that is, if you have some bulk data for these proteins from previous studies of synchronizing the cells, how does the magnitude of the response that you measure in single cells compares to the population averages from previous bulk studies?
Okay, so to address the first part of your question, I mean, clearly, this would be a great experiment to do with an unbiased approach like a single cell mass spec. We are stuck having to use antibodies, so we have to define the set of features that we want to look at. And to be honest, this set of features is just informed by our current knowledge of the cell cycle. So the things that we know are involved in cell cycle progression. And we tried to choose features that regulated different parts of the cell cycle so that we could really get a good coverage of all of the regulatory events of the cell cycle. So that's basically how we chose this feature set. I would love to redo this experiment using an unbiased method like single cell mass spec at some point. Of course, when you have an unbiased method, this opens the door more for discovery of novel regulators of the cell cycle, of things we didn't know about. We are unfortunately stuck with looking at things that we think might be involved and hoping to identify novel behaviors or novel interactions or novel mechanisms among these known cell cycle regulators.
And that's very valuable. I think the two approaches are quite complementary, and in particular, I think it's great to measure the same proteins with different methods so that one can cross-validate and have higher confidence. But in this case, so it was a combination of looking at multiple previous studies and proteins that are involved in regulating cell division. Did you see that one expectation that they would have is that perhaps the dynamic range that you're able to observe in single cells is larger than the dynamic range seen in bulk studies, because there we would have certain degree of averages and we would not see the cells with the most extreme levels of the protein. Do you see that?
Well, I would say that, I mean, one of the things that would be obscured, it's sort of along these lines, one of the things that would be obscured by bulk population measurements is this phenomenon that we see with replicative stress, where we actually induce two distinct states of arrest in the same population. If we were taking bulk measurements of after a toposide treatment, we wouldn't be able to distinguish these two phases. And these two phases have distinct signatures, and this would be all obscured if we tried doing this by a Western blot or by some other bulk mass spec measurements.
Mm-hmm.
So they would just look like the blending into one state. So that's really the power of using a single cell approach for a question like this.
Yeah. And one last question. I have many more. I just don't want to monopolize you. We can talk more offline. But one last question is, on some of your diagrams, some of your topologies, it was very clear that there is just a very thin streak of cells that express a certain protein at high level. For example, E2F1 was one such example, where you have cells that seem to form a highly coherent cluster, and within that cluster you see just a very thin streak of cells that have much higher abundance of E2F, and their neighbors do not have that. How do you think about this phenomenon? Do you think that all these cells forming the neighborhood are very similar to each other in the abundances of all proteins except for E2F, or all features that you measured? You used a lot of different features in this case, and just E2F is different, or what's your interpretation of seeing this? I think it's interesting. I'm just wondering how do we interpret it?
I think that's exactly right. I think that other than E2F, the rest of the signatures of these cells are identical, right? Are very, very close to those cells with high E2F. And also in that region is where all the cells with high E2F are. It's where it's a hot spot of it's S phase. It's where E2F is highest. It just happens that there's a subset of cells that are expressing a lot of E2F. So our dimensionality reduction method, where FATE is not distinguishing them as a distinct group of cells, subset there, but it is possible that they are. It's something to look into for sure.
Do you know what fraction of the total variance in your data is captured in the lower dimensional projection? That's always a trade-off. The lower dimensional projection helps us understand and visualize the data, but do you feel that it really captures all of the essential dimensions, biologically meaningful dimensions in your data? Or do you think that perhaps there might be additional features that are just hard to project with any method?
Yeah, so I mean, that's a very good point. We have not calculated what amount of the variability we get in these lower dimensions. I can say that we have, we of course have we've projected these into 3 dimensions, and I haven't shown any of these structures here today. There seems to be a bit more texture among those 3-dimensional structures. In fact, actually, we used the single cell VR approach that Luca presented yesterday to explore the 3-dimensional structures we see here. And there's a bit more texture to those structures, but we don't see the appearance of like a novel cluster of cells when we increase up to 3 dimensions. So we could be missing some information, important information at these lower dimensional structures, but I think comparing the 2- and 3-dimensional structures, it doesn't feel like we're losing much information, at least at that level. So I'm relatively confident that we're not missing something extremely important.